Hi viewers, welcome to Capsule. Today's topic is opsonization. So opsonization is an immune response which uses opsonins to tag foreign pathogens for elimination by phagocytosis process with the help of phagocyte cells like NK cells. Okay, so we can write like an immune process in which opsonins eliminates pathogens by phagocytosis process. Because without opsonin, such as antibody, the negatively charged cell walls of the pathogen and the phagocyte cell will repel each other. So, opsonin are need to bind with the pathogen. Now we will see how this process occurs. So, this is a pathogen which is detected by specific antibodies. This segment is FAB or fragment antibody binding, antigen binding fragment, and this is complementary fragment or FC, which will attach with the FC receptor present in the NK cells or phagocyte cells. Okay. If you don't have any clear concept about antibody structure, I had a video also in phagocytosis process. You can go through it. Okay. So after detection by the antibody, this antigen antibody complex will attract the NK cells. This is a kind of phagocyte cells, okay, or natural Kupffer cells. And now these NK cells will form a complex with this complex. This is FC part. And this is the pathogen, okay. And after attachment, these NK cells will release a cytotoxic vesicle which contains granules to destroy the pathogen. Okay, release of cytotoxic granules or content, which will destroy the pathogen. So this is the destroyed pathogen, and the death of pathogen will occur by necrosis process, or it can be membrane damage process. So that's why this process is also known as enhanced attachment, because which results in much more efficient phagocytosis. Now we will see which kind of opsonins or antibodies are used. Okay. These are IgG kind of antibody, IgM kind of antibody, or it can be proteins like C3b protein, C4b protein, etc. And you should keep in mind that this process actually acts on fungal pathogens through their fungal surface exposed ligands. Okay, so this is the optimization process kind of antigen antibody reaction. This is the end of my lecture. If you like my video, please do like, comment, share and subscribe. Thank you for your attention.